Welcome to the Genetics Podcast. This is another episode of our Precision Pioneers miniseries, and I'm excited to be here today with Dr. Oscar Segurado, who is the Chief Medical Officer at ASC Therapeutics, which is developing gene therapies for rare disease using CRISPR. Oscar's a medical doctor, and he's worked on the cutting edge of precision medicine for essentially his whole career, so I'm really excited to have him here today and to be able to pick his brain on what the future of personalized medicine might look like. So thanks so much for joining me.
Thank you so much, Patrick. I'm really excited about this conversation, and I know Sana Genetics. You have a great team there. I've been interacting with some of them, so happy to be here in your podcast.
Great. Thank you so much, Oscar. I'd love to actually just jump right into talking about CRISPR and its use as a therapeutics tool, since at ASC you all are working right at the cutting edge here. Could you just explain to anyone who's not familiar what CRISPR is and how you all are applying it to treatments for rare diseases?
Thank you, Patrick. So I think it would be useful if I start telling you a little bit about myself and the company I'm working for.
Great.
So I'm a physician scientist. I've been working in clinical development and translational medicine, meaning bringing lab work, of course, genetics, genomics to the patient bed. And in the past 10 years, I've been focusing on cell therapy and gene therapy. My background is in immunology and cell therapy and molecular biology. And I joined ASC Therapeutics in 2019. Originally, this company was built as a life sciences manufacturing provider for biopharmaceutical companies. And the company derived some of their work from interactions with the Stanford University. And at the Stanford University, as you can imagine, they have been working on CRISPR and other gene editing technologies for many, many years. One of these technologies is called Target. And Target was the origin of the original company, Applied Stem Cell. That's why we're called ASC Therapeutics. And this proprietary technology is the basis of working with other biopharmas, where we put together line cells, animal models, and in addition, while we were already working on that, practically is when the CRISPR field exploded, and we in-licensed that from our colleagues at Berkeley University up the road in some way. And with that work, we started helping others. And at some point, the company decided, okay, now we want to start developing therapies ourselves. We want to deploy our know-how on CRISPR, on Target, which is this other technology that I mentioned, and also the standard gene therapy technology for different diseases. So they started incubating about 4 years ago this kind of work. And that's when they brought me in. And I am extraordinarily lucky to be with a company that is really growing very, very rapidly. And indeed, as you mentioned, we are working with CRISPR technology to support rare diseases. And I would be happy to explain that to you. That's just the kind of the basic understanding, the history of where we are right now.
Excellent. Yeah, it would be great to dive into an example of where you all are applying CRISPR to treat rare disease. And I'd also love to learn about some of the other technologies that are maybe not as mainstream, but certainly stem cell and cell therapies in general are an enormous field. So maybe we can start talking about CRISPR and then talk about some of the cell therapy side of things as well.
Yeah, so CRISPR is a technology that allows us, the world, the scientists, to manipulate any gene and being able to either replace it or make some changes on this gene that make it functional, especially when is either deleted or is non-functional. And that's what happens with a disease that I'm going to give you as an example, which is the one we're working on. The disease is haemophilia. Haemophilia is a disease that happens when any of the clotting factors that are required for blood coagulation is not working. And there are two major clotting factors that are missing in patients with hemophilia, either factor 8 or factor 9. And we have started working on factor 8, although we have the same technology and we plan to use it with factor 9. when an individual has a failure on factor 8, the cascade of clotting factors, because this is the way the blood clots, does not work because something is missing. So you need to replace that factor 8. And that's where we started working maybe about 5 or 6 years ago, first in animal models. And we started replacing in these mice that had already a deletion on this factor 8 and replacing it with CRISPR. It worked. So it was published in Nature. And with this information, we started working on a next generation of a factor 8 replacement gene. So in general, the way it has been done, because this is a work that started maybe 20 years ago, I mean, any kind of gene manipulation for helping patients with hemophilia A, and hemophilia A is the one where there is a missing factor 8. Everyone has been using a factor 8 that is basically the same as the regular human factor 8. What differentiates our work is that we decided to in-license work that had been done at the Emory University and a company called Expression Therapeutics, where they created a chimeric porcine human gene. So you would say, okay, you are mixing pig sequences with human sequences.
Yeah, what could go wrong?
Everything is going to go wrong. That's the first thought that anyone can have, except that Emory University had been working on something called ancestral sequences of factor 8 for almost 3 decades. And part of that work became one of the most successful factor 8 replacement therapies. It's called Obisur. It was in-licensed by Takeda and is still one of the top replacement therapies. And by the way, this is almost entirely porcine sequences. So I'm giving you a little bit the story, which I think is very important. I never had really an opportunity as I have today to explain this, because what they figure out with these decades of experience and this developing Obisur, again, branded FDA-approved factor 8 for patients with hemophilia A, is that they realized that there were certain sequences that allowed the factor 8, when it was incorporated into any cell, to be more efficiently processed. It's called biosynthesis, and more efficiently secreted from the cell. And because everyone is now very familiar with mRNA vaccines, I can tell you that there are several similarities here. So when I talk about this, because now it's in the mainstream media, you're going to understand that when you get a vaccine, you get the mRNA, everyone knows, messenger RNA, it goes into a cell and it starts producing proteins that are the ones that are very similar to COVID-19. In our case, we don't provide the mRNA, we provide the DNA. So, and this DNA is transcribed into mRNA. So it's a kind of a step earlier. And that's also what happens with other vaccines like Johnson & Johnson. So, but we do that. That's the way we treat these patients. We provide them some DNA. This DNA is integrated into the chromosomes of the cell, and it starts producing mRNA, and the mRNA produces protein, which is factor 8, and is secreted to the circulation. Okay. Because we use these porcine sequences from the pig, which is 9% of the entire gene that is called transgene, this protein is much more efficiently processed, biosynthesized and secreted than regular human proteins. This is the work that we have been doing over the past, especially since I joined the company in 2019. And now we are ready to present all the data that we have produced in animal models, in 3 different types of animal models. It's called wild-type mice, humanized mice, and also with primates, with monkeys. This data is now put together in a package that has thousands and thousands of pages, and is going to be submitted to the FDA in the next few months. So I expect, because my role, I'm Chief Medical Officer, my role is conduct clinical trials, but I mean, I need to know a little bit more than that. And we are planning to start putting this transgene, this chimeric transgene, into patients sometime this summer. This is going to start what we call a phase 1/2 clinical trial that is going to look into safety, is going to look into the efficiencies of how we are able to produce this factor VIII in these individuals. We're working with some of the top experts in the field, especially Professor Stephen Pipe from the University of Michigan, and he totally endorses our approach.
This is fascinating. I have 20 questions at least, but the first one I want to ask is, where are you actually inserting the gene? Is it into blood stem cells so that it can then produce many more healthy cells? Because I think that's probably one of the big challenges here, right? Of where do you actually make the edit so that you don't have to continually edit over and over?
Excellent question. And guess what? As I mentioned, we in-licensed factor VIII from the Emory University and a spin-off company called Expression Therapeutics. So what we licensed from them was the potential of using not only the transgene, but also something called a promoter, which is the one that really makes the protein the production, when we do the gene therapy. Excuse me, in case of gene, we are still in CRISPR treatment. So there we are really only using the transgene and using a promoter already in the genome. When we do that, we are inserting the gene, which was your question, into a hepatocyte, the chromosome of a hepatocyte, which is a liver cell. So we use the cells of the body, in this case, a liver cell, a hepatocyte, as a biofactory. It's a chemical factory. So it's the one that is going to take all this information and start producing everything. When you take the COVID vaccine, an mRNA, you use any cell, usually cells in your muscles, and they are going to present because there is no release. So these spike proteins from the COVID are expressed in the surface of the cell, and the immune system recognizes that and is going to start producing antibodies and T cells that are going to create the immunity. In our case, we release factor VIII, but the process is similar. Okay, we are using a cell as a biofactory. Now, this is what we are doing, but the company that initially licensed to us the therapy is also using the exact same chimeric transgene in stem cells. So the answer to you is that you can do that both ways. So we are doing that in two ways. We are using CRISPR, and we are also using what is standard gene therapy, where we are going to provide the transgene plus a promoter, and I can explain that in a moment if you want to see the difference between CRISPR and gene therapy. And again, we use these cells that are the ones that are going to start producing this protein. Now, it doesn't matter if you use stem cells or the hepatocyte. At the end of the day, you are forcing one cell to do something that is not used to do. Because even if you think, and this is real, factor VIII is usually produced in the liver. People that get a liver transplantation with haemophilia, boof, from one day to another, they're cured. So, but we know that production is not happening in the hepatocytes. So we are forcing a cell to do something that is not used to do. The hepatocyte produces hundreds and hundreds of proteins. Again, it's a huge chemical factory with the assembly line and everything there, but not this factor VIII. So that's why everything that we're doing is going to help the hepatocyte be more efficient producing factor VIII.
That makes complete sense. I would love if you could actually give a little bit of a high-level taxonomy of gene therapy, CRISPR, stem cell therapy, how do these, they're overlapping, semi-overlapping categories sometimes. You could give a high-level overview of, because I think you all work across a number of these areas, which is relatively unique. And I think because of the way that ASC started and has now moved into being a therapeutics company is probably the part of the reason for this, rather than someone arriving with a new technology or molecule and then being laser focused on that. So yeah, if you could, I think that would be great.
Sure, glad to do that. And at some point, I was able to achieve a tenure professorship in the University of Lyon in Spain, which I still maintain, a little bit remote in this case. But I like to teach, and I can tell you the way I teach gene therapy and gene editing so that everyone can understand it. And I use an analogy. My analogy is Apollo 11, is how we were able, the humanity, to get to the moon. And I think this analogy works because we needed a number of elements. Let's start with this. This is
for those who can't see, he's got a plastic rocket on screen. So
it's a plastic rocket. You won't believe where is this plastic rocket coming from. It was a guy that was also seeing some slides that I used for this analogy. And he said, hey, look, I have a 3D printer and I have already a rocket and a module and everything. So you're going to see more pieces that he sent me over the mail for free. So this is the rocket. And the rocket is the virus. We use an AAV virus. It's an adenovirus. And this kind of virus is totally harmless. And what it does is that it has tropism. We use one that is called AAV8, serotype 8, which has tropism for the hepatocyte. So we have all the information in this rocket, which is what the patient receives as an infusion. Now, this rocket finds the hepatocyte, goes into it, gets into the nucleus, and now imagine that the chromosome inside the nucleus is the moon, okay? So there are two ways to deal with this. This is now the other piece that the individual sent me. And this is the capsule. I call it more the lunar module. And here is the promoter. And by the way, they can be separated, okay? So this is what we put around the nucleus, around the chromosome, inside the nucleus. This is DNA, and this is a piece of information that tells this DNA to start producing mRNA, okay? So when we use gene therapy, these two elements are orbiting the moon. And the promoter tells the transgene to start transcribing and producing mRNA. This mRNA goes into the endoplasmic reticulum, Golgi apparatus in the cytoplasm, and it starts producing factor VIII and releasing it. This is standard gene therapy. Now, let's go to CRISPR. When we use the CRISPR technology, we don't have this. We only have this, okay?
So it's just the DNA, not the promoter.
It's just the DNA. But we need to do something else. We send two rockets. The first rocket has the CRISPR-Cas9 machinery. And what it does, and I don't have, unfortunately, anything to show you here, is that it makes a cut. It has something called guide RNA. So it knows where to go into the chromosome. So it goes now into the moon, which is the chromosome. It identifies a very specific concrete site that you want to manipulate. And it has attached to that Cas9, which is an enzyme. And this enzyme produces a double-stranded cut. Boom, a cut. This is what we call also knockout. Now, the second rocket has now this DNA. It now follows practically the lead of the previous rocket and lands in the moon. This is the lunar module now.
Right. And you set up a colony and never leave, right?
We even open us a little bit here.
Perfect.
I don't know how these 3D printers work, but it's amazing. So
pretty impressive.
Now you are on the moon. You are inside the chromosome. And since you don't have this, which is the promoter that is telling you how to start producing protein, we locate it in a very specific site that requires a lot of work that we have been doing to know exactly which exon in which area needs to be, so that we have another, it's called another promoter. It's called safe harbor promoter. And the one that we use is for albumin. Albumin is a protein that is continuously measured in blood. Is one of the most efficient promoters we have in the body, because albumin is an essential protein that we need for everything to function. So we put that DNA coding for factor VIII close to the existing promoter for albumin, and this factor VIII starts to just work like before, producing an mRNA for factor VIII, protein for factor VIII, and secreting factor VIII. That's the CRISPR-Cas9 machinery.
What a great metaphor. I love that. That's going to stick with me forever. So really what it boils down to is the DNA being integrated into the genome, at which point you've really latched onto the albumin promoter and the part of the genome machinery to allow the DNA, which encodes for the protein, to continually be produced, versus in the gene therapy approach. I love this idea of it's orbiting. So and I guess in that case, you've got to, generally speaking, continuously reinfuse the gene and promoter, right? Because it only works for some time, or is that not the case?
This is an excellent question, which is still open. There are many companies that, believe it or not, we are a very tiny company compared with Pfizer, Roche, Biomarin. They're working with this fully human gene on this gene therapy. So we think that we can maintain and make this protein production more durable, because that's exactly the point. The problem is, first, since you are not integrating the gene into the chromosome, landing on the moon, if this hepatocyte divides, you lose everything. You don't have anything. And that's what happens with children. That's why this therapy right now by everyone in the field is only useful. I mean, not gene editing. I'm talking about gene therapy for patients that are 18 years or older. Now, gene editing, CRISPR, because you are integrating the gene into the chromosome, if this cell divides, like happens in children, because
your liver's growing,
the liver is growing, no problem. That's going to work. So we're going to be the first to be able to look into what is called in vivo CRISPR therapy for patients with hemophilia A.
That's fascinating. So why did you all choose hemophilia A as the first focal point? I assume there's probably the sky's the limit in terms of the other liver-based and potentially also blood-based disorders where the proteins are produced in the liver. I know you're not always able to talk about where else you're thinking about, but I'd love to hear why start in hemophilia and any thoughts on what else is out there in terms of opportunities.
When we speak with venture capitalists, investors, and private equity, and so on, and tell them what we're doing, they say, "Are you crazy? So you're working in the most crowded space in the field?" Exactly what you said. The reason hemophilia A is so relevant is because, again, the first work started in almost two decades ago. The first patients were treated 5-6 years ago. by some of the companies I mentioned.
With gene therapies.
With gene therapies. Gene editing, no one has ever done it yet. So we're going to be the first. But we thought that because of all this amount of information, we are continuously, even yesterday I attended a convention, and it was called Gene Therapies in Blood Disorders, and about half of the meeting was devoted to haemophilia. You learn a lot. You have already all the information there. So we have examples, we have publications, we have a specific guideline from the FDA on haemophilia. So we are going into a field that has already such an amount of know-how that we are going to refine it. So that's why there are so many copycats now from Tesla, because they put the ideas out there, and now there are 100 different electric vehicles out there. I don't think that's a problem. I think that's, again, how science and how biopharmaceutical companies work. So we feel comfortable with that. Now, there are several other areas that have been very successful, and I've been part of that. I've been working with Avexis. It's a company that was focusing on spinal muscular atrophy, the SMN1 gene, and the company was acquired by Roche for close to $8 billion, and now is one of the most successful therapies because it treats these patients with gene therapy, and the therapy is called Zolgensma. So I was part of the company for a couple of years. So I think that and we shouldn't forget this. This is the start. We are in the earliest stages of gene therapy, gene editing, and all that. Now, there are several companies, CRISPR Therapeutics, Editas Medicine, there is Intellia, there are Sangamo working also with an alternative to CRISPR, zinc nucleases. So zinc finger nucleases. So there are many different companies working in different spaces, but I think that's the explanation for hemophilia A. And we're a small fish in a small pond, I would say too.
Yeah, but I think as you say, the tidal wave is coming, right? It's only the beginning. What are the general, so moving beyond hemophilia, what are the general characteristics of a disease for which gene editing is a viable approach for treatment?
In general, all these advanced biologic therapies have an intrinsic benefit of being precise tools for manipulating human biology that you understand. So that's the first thing that needs to happen. Right now, there are over 5,000 genetic diseases that we can call monogenic. So there is one single gene that fails for whatever reason. And in many of them, we understand the biology. So those are diseases that we can target. Now, there are about 100,000 genes that are associated with common diseases like diabetes, heart disease, and so on. But they are multigenic. So you really cannot try to spend your money, your time on making any kind of gene therapy or gene editing, because you don't know what's going to happen. But in cases like Avexis I mentioned with SMN1, we knew that if we were able to replace that protein, these children that usually die with 1 year or 2 years of age are going to live forever. And this is what is happening. Some of them are now, I think, close to 8 years old. So we with hemophilia is the same. This is a disease where it's one single gene. We know that we need to replace factor VIII. There is also a number of diseases in the central nervous system, also in liver related monogenic diseases that we're also working on. The industry. So we have several ideas. There is also sickle cell anemia, beta thalassemia, where the changes that you make are in stem cells. You don't touch the liver. You take the stem cells from the individual, you manipulate them and put them back into the individual, and they start producing what is missing in sickle cell therapy. There is also, you need to get the right size and form of the red blood cell. So we are very targeted on diseases that we know especially from experimental models, animal models, that we are able to reach a positive result.
And I guess the delivery is a big challenge. If we think about neurology, for example, in a disease like ALS, where the genetics is starting to be really well understood, but delivering the rocket, using your analogy, to the brain is still a big challenge. Whereas in blood stem cells or the liver, you've got a much easier route to get to the tissue.
Absolutely. But again, with ALS, that's one of these diseases that probably multigenic. And that means that it's probably going very difficult to, although there might be some patients with ALS that have a single genetic mutation and you could treat them. So you first need to also do all the genomics that you guys do very nicely at Sana Genetics and identify those patients, where is the mutation, and then see if those patients are really the ones that you can treat with either gene editing or gene therapy.
Yeah, that's completely clear. So you mentioned earlier that as a chief medical officer, one of the biggest things you're thinking about all day is how to design and run clinical trials. And I'm interested in what are the challenges that you face using a very new technology. I think, as you mentioned earlier, you all will hopefully be the first to do this with integrating into the genome. And it sounds like there's a group of patients, including young children, where this will be totally unique. What challenges do you face in designing clinical studies in these examples where there's no existing therapies? The route through the FDA is often not particularly clear, although a little bit better trodden in hemophilia A. I'd love to hear more about how that differs from your past experiences with maybe more well-trodden clinical paths.
So great question, and I can tell you multiple challenges, but we have some advantages. Number one of all of that is that the FDA has opened their arms, and they're even asking all the, let's call it, competitors to work together. They have sometimes even 2-day seminars where they bring all the companies and they start talking about all these challenges. Now, again, I've been working with, for instance, with the Humira team. At that time, it was monoclonal antibodies. It was something relatively new. It was a biological therapy. We were coming from small molecules. And when I was working with Abbott AbbVie for over 15 years, I really learned a lot in terms of clinical development. But so all these learnings, at the end of the day, the clinical development hasn't changed. So you first have to do your phase 1/2. You have to do a dose ranging study. What dose is the adequate for the patients? Then you move into the so-called pivotal trials, where you demonstrate that indeed your drug, your therapy is safe and effective. That hasn't changed now, and I'm doing that, following the book in terms of the typical standards. Now, there is something that is unique to gene therapy and gene editing, and I was not aware of that really when I joined first Avexis and then ASC Therapeutics. And this is that even as a chief medical officer, I have to understand key aspects of manufacturing, analytics, and even what is called P&L, which is understanding the way you produce the drug and the way you deliver it. So it's the kind of the supply chain from the place where it's produced until you really put that in the patient. And this is extraordinarily complex. I am involved because when you are in a small company, if necessary, you also change bulbs. I'm involved directly in the manufacturing decisions. Obviously, I don't do that directly myself, but we're looking very carefully into our CMO, the company that is doing all the manufacturing. And because of our previous experience as a life sciences manufacturer, we are able to also be completely involved. We are planning to develop a pilot manufacturing facility in our headquarters, and also start having a larger scaled manufacturing facility over time. But this is something that is extremely complex. The way you put together this,
the rocket,
I can tell you the rocket is extraordinarily complex. This has nothing to do with putting together a monoclonal antibody which chose cells. That's the way we do that. We use different cells. We use 3 plasmids. They need to be cooked with a special recipe. It fails many times. So you need, just to get the material that you need for one single patient. The first patient that I'm going to be treating sometime this summer is going to take us weeks and weeks, if not months of work for one single dose. And the average cost in the industry for one single dose, standard cost, is close to $1 million.
Wow. Which is, I think, a great segue into one of the biggest challenges in rare disease, which is the cost of producing, and then ultimately, as a result, the price to patients' healthcare system. It sounds like you're starting from, the manufacturing costs. As you go up, the price will go down, or the cost will go down, but it's not going to go down to cents like it might for a small molecule. How do you think about the industry challenges as a whole in balancing these transformative therapies with the incredible costs, and what are the levers that we have as an industry to help balance the books a little bit better?
Good point. I think you have noticed by this point that I like analogies. So let's go back to the electric vehicles. So the first Tesla, the sports car was $150,000. Now they're mentioning that the next one, the first is going to be $20,000, but there are companies already out there announcing, and they have electric vehicles for about $5,000. So that's going to happen. So it's a matter of time. Now, we are not worried about having Zolgensma, the drug that I was working on with Avexis. Right now is $2.1 million. The company was acquired by Novartis. And is working very well. So there are many countries, and the most of the healthcare systems are paying for it. Why is that? Because you tell the parents that are very happy with their child, and they are telling them your child is going to die within 1 year. So you're going to find that money, and the system is going to find that money, because you're going to make this kid having an absolutely normal life. And I just read a few weeks ago that the youngest child was treated with Zolgensma, and I think it was 2 weeks old. Okay, so yes, that's the cost, and this is an issue. We will have to work on that now. The cost is going to go down. There's no doubt about that. Now, but let me go to the haemophilia. In haemophilia, you have these patients that need to get replacement therapy weekly. And the yearly cost for these patients, counting with everything, including the potential for several types of side effects and problems associated to that, could be around, I think, the average in the US, obviously I'm speaking about, $250,000 per year. So you just multiply. In 4 years, 1 million. In 40 years, 10 million. So this therapy is going to be very costly. Now, no one knows how is it going to work in the United States. If you use the American system where there are health insurance, and this health insurance don't know if you're going to be with the company 2 months or 2 years or 20 years, why should they pay $2 million for a hemophilia therapy that is going to benefit another health insurer? So we're, as an industry, and the entire healthcare companies are working on that and figuring that out. Now, this is not a problem in the majority, I would say, of the Western countries, Canada, UK, the entire Europe, and many other countries around the world that have universal health care. In that case, there is no problem. They are going to do their math. They are going to calculate, and they say, okay, I have to maintain this person anyway, because this individual is 20 years old, and in general, the average life of a hemophilia A patient is going to be, I don't know, up to 70 years. So I have to pay for all these costs for 50 years. Now you're telling me that the company is going to give me a one and done, one single infusion, these couple of rockets, and everything is going to be done in 30 minutes. And it's going to cost me $2 million, or to the healthcare system is going to cost $2 million. They're doing it. So they're already considering it's happening.
Yeah, I think it's such a great point. The one concern that I have is whether the pace at which the cost comes down, if that goes more slowly than the pace at which smart companies and scientists like you start inventing new therapies, we may end up with the case which is a win for patients, in which many of these 5,000 rare monogenic diseases that don't have treatments all of a sudden have treatments, but they all cost $2 million. And at the moment, the healthcare system just isn't, with the exception of things like haemophilia, paying for it. There are other cases where patients are just simply, dying at a young age, for example. And then you have this net positive clearly for the world and for patients, but the healthcare system can't cope. I'm wondering if can you give me a layer of optimism that the manufacturing costs will start to come down quickly enough that this won't be a problem?
This is not my field, manufacturing costs and so on, but I can tell you that we are all seeing that is happening. It's going to happen. Now, I think that the system is going to adjust to that. It's going to adapt. And I can tell you the health insurance are smart enough, and they're not going to go bankrupt. So they have their stockholders out there. So they're simply going to deny therapies for other people, and they're going to tell them, okay, stay with your current therapy and so on. So many patients, and we're encouraging some of them, are joining studies. The BioMarine study recently included 300 patients. So all these 300 patients are getting it for free. And once we finish our phase 1 clinical trial, we're going to have also another 300 patients that are going to get our therapies for free. So everyone is going to see things going their way, and everyone is going to adapt to that. I'm not concerned with costs. We're working on it.
Great. Yeah, it does seem like certainly all the incentives and the market pressures are there that these contract manufacturing organizations and technology developers it's high on a lot of people's lists, I think, to figure out how to get these things down by orders of magnitude. We're coming up close to the hour here, so I wanted to maybe just finish with another high-level question. You've worked at the front leading edge of precision medicine for basically your whole career. I'm curious what obstacles you see in place to us living in a world in 10 years, 20 years, 30 years time, where everyone has access to precision or personalized therapies. I'd love to hear, A, what that world looks like to you, and B, what are the critical things that we need to accomplish in order to get there?
So let me start saying that gene therapy, gene editing are the culprit of personalized medicine, because first of all, these are rare diseases. We're really pinpointing into a gene that is not working, and we're replacing and we're curing it. So this is not a therapy. This is a cure in many cases. So this is the ultimate personalized medicine in some way. Now, I mentioned at some point the hepatocyte, that we're using the hepatocyte of individuals as a chemical factory. Okay, so because of my background and experience in genomics and personalized medicine, precision medicine, I am also working, which is going to be different from other companies working with hemophilia A gene therapy, on ensuring that we identify all the critical elements of the hepatocyte of each and every patient that receives our drug, so that we personalize the therapy to that individual, and we start understanding which individuals are going to respond better than others. That's one of the problems that we have seen in hemophilia A, and this is that there is a large variability. Some patients, and nobody knows why, do not respond. So the rocket stuff and the orbiting capsule and promoter are not doing their job. With other people, it works perfectly. So there is a large variability, and nobody understands that. We are going to look into that. Our company, especially under my leadership, we're going to look into a number of ways to look into how this liver of this individual has been working over the past several years, retrospectively, we're going to look if there was any kind of infection, liver infection, especially hepatitis B and C. And we're also going to look into the way the function and the structure of the liver works. We're going to do ultrasound every 6 months. We're going to look into a number of biomarkers in collaboration with Siemens. They have developed some biomarkers that indicate the potential of fibrosis. So we are going to follow up each and every patient in our clinical trials as it has never been done before. So that's why I like to talk when I have speak engagements. I talk about gene therapy and precision medicine. So I'm combining both, and I think that that's extraordinarily important.
Yeah, that is. And this issue of variability, both in presentation of disease course and symptoms and response to treatment in rare disease, I think is a big challenge, because we know that this is the case in common disease from numerous studies, because there are enough people. We know it's the case in rare disease anecdotally, but the numbers are often too small to really understand in a statistically rigorous way why is it that that patients with this already rare disease actually are rare subtypes. So I think the approach you describe is the right one, whereas is to gather as much information as you can and follow as long as you can to really, because when you have the patients that are going through the incredibly rigorous clinical program that you've already got going, and you've built this base of incredible data from the start, it makes sense to get as much information as you can out of that. Otherwise, someone will have to go back and do it again from scratch in an academic lab, and it's going to cost millions of dollars all over again that could have been avoided. Just want to say thank you so much. This was an incredibly enlightening interview, and in particular, I think you're probably the best in the business at explaining some of these really complex concepts using accessible analogies. So thank you for that. I think I'm going to reuse the rocket analogy certainly in the future, and I'll give you due credit for it. So thank you so much. I really appreciate it, and I hope everybody checks you out on social media, LinkedIn, Twitter, and what is the website they can visit if they want to learn more about you all's work?
I would say that is www.actherapeutics.com, and they can also Google my name, and I also they're going to see some other aspects of my work.
Great. Well, thank you so much. And it's Friday. Well, it's Friday morning for you. It's Friday evening for me here in the UK. If you were in Spain, you would be you'd be done. But I think the pandemic has prevented travel back. But thank you so much, and look forward to talking again soon.
Thank you so much, Patrick. Great pleasure speaking with you today.
Thank you for listening to this episode of the Genetics Podcast. If you enjoyed the podcast, we'd really appreciate if you left us a review on your favorite podcast player, or even better, you can tell a friend who you think might like it too. As always, you can reach us anytime at podcast@sonogenetics.com. We really love to hear from you all about any feedback you have, guests you'd like to hear from, or topics that you'd like to see us cover in the future. Thank you again for listening, and we'll see you next time.